about chapter 21 and evolution. So, a lot of this stuff, again, things that we talked about in honors biology, so hopefully some of it at least rings a bell. So, when we think about evidence for evolution, usually what comes to mind is Charles Darwin and those wonderful Galapagos Islands and his group of 14 finch that he looked at and he collected specimens from. So all of those 14 finches were very similar, except they had different beaks. So, well, why? Why would they have different beaks? Well, he figured that they must have different beaks so that they could eat different types of food. And so apparently he wrote, one might really fancy that one. Apparently he liked some of the beaks better than others. One might really fancy that one species has been taken and modified for different ends. They wrote funny back in there, but back in the day. But of course, what he means is, well, this beak can be used for little seeds. This beak can be used for big seeds. This beak can be used to maybe eat insects even. This finch, which I think he's pretty genius, he actually was able to use some kind of a little stick, and he could get down inside of a tree bark and get out larvae from insects. So he was pretty smart. That's how he used his beak. And there are ground finches that, if you notice their beak really substantial, they were able to eat larger seeds and crack those open, whereas this poor little guy would never be able to crack those seeds open. And, all these different finch for all the different types of things that they could eat. And that's really all of these pictures, what they show. This is a picture that I kind of like because it shows this is where the ancestral original finch would have been and what all evolved, what all characteristics evolved from that finch. Not only shapes of the beaks, if you take a look at all the beak shapes, but also size of them. This little finch was a little guy. Why was that? Well, apparently size, that small size was advantageous to where they lived. The other reason I like this is it shows you all of these beaks. Well, they were for eating insects. These beaks for eating seeds, especially look at that guy, nice big seeds. Cactus seeds and other cactus parts, these two, and then buds and fruits. He's definitely got a way different shaped beak than others. So that was one of the first things that we used for evidence that evolution or change over time has taken place. So we mentioned this in chapter 20 as well. This is just a repeat. There are 3 things that need to happen in order for natural selection to occur. There has to be variation. That variation has to lead to individuals being able to reproduce successfully. And then obviously these differences have to be transmitted genetically to the next generation. After Darwin, of course, lots of people take what one scientist did and then they study them even more. There was a couple, Peter and Rosemary Grant. They actually just looked at the ground finch, and they even found that within one species of finch, there were different variations. So they looked at the beaks of just that one particular kind of finch and found that the average beak depth changed from one year to the next. And that was because of droughts and normal rains. So even within 1 year, within one species, there can be differences. So during drought years, the birds with the deeper, more powerful beaks survive better. Their numbers would increase. And then in the next year, if there were just normal range, then the average beak depth decreased to whatever its original size was. And again, that's what all of these pictures show you. So when environment changes, natural selection often favors different traits in a species. If you think back to honors biology, we actually talked about this already. We talked about the peppered moths of England. And if you remember it all, there are light moths and there are dark moths. And we talked about what happened during the Industrial Revolution. So, if you remember this, before the Industrial Revolution, the trees had very light bark. And so here's a light colored moth and here's a dark colored moth. Well, obviously, if you were a bird and you were hungry, what are you going to see first? Well, you're going to see the dark colored moth. And so before the Industrial Revolution, most of the moths, I think it was 90% of 80 or 90% of the moths, were light colored and only 10 to 20% were dark. Well, Industrial Revolution happened. We started burning a lot of coal and other fossil fuels, and all of that soot covered the tree bark. Well, at that point, then the dark moths were actually they were and it had an advantage because they were able to hide against the bark on the soot covered trees. And so now, if you're a bird, you see this light moth first and not the dark moth. And so their population numbers flip flopped. There were going to be 80 to 90% of the population were dark, and only 10 to 20% were light. Kettlewell is the one that we usually talk about with regard to these moths because he's the one that actually did some testing on the hypothesis. Dark tree trunks, more dark-colored moths survive. Light tree trunks, more light-colored moths survive. And so when environmental conditions reverse, so does whatever selection pressures there are. We refer to this as industrial melanism. It's a phenomenon in which darker individuals come to be predominant over the lighter ones. Now, obviously, since then, that all happened in the 1800s, we've tried to control pollution, and this has resulted in lichens growing on the trees and the bark color becoming lighter again instead of dark. So now we've got, once again, more light-colored moths than dark-colored moths in this day and age. And that just shows population trends, the agent of selection. Sometimes it's difficult to pin down. Sometimes it's pretty easy to pin down. And the moth selection was an easy one. Fossils are also evidence of evolution. You know that fossils are either the preserved remains of once living organisms or they are impressions of once living organisms. Rock fossils are created when three events occur. Organisms become buried in sediment, the calcium in the bone or whatever hard tissue there happens to be in that organism turns into minerals, and then the surrounding sediment hardens over it and around that to make it into rock. And so when an archaeologist or whoever's doing a dig, a geologist comes across a fossil, well, they want to know how old is it? And they can do what's referred to as absolute dating, which is determining the age of fossils by their radioactivity decay. And we'll talk about that in just a minute. They can also just look at the position of that fossil in the sediment and do some relative dating. So, of course, older fossils will be deeper down in the dig. Younger fossils or earlier fossils that haven't been around that long will be at the top of the dig site. There are lots of different isotopes that we use for absolute dating. Uranium-238 is one of them. You have probably heard of carbon dating. And what we need to understand about all of this process is something known as a half-life. And a half-life is the rate of decay of whatever that isotope in the organism is. And so this is probably not going to make any sense to you until you look at this video, and you only need to watch the first 2 minutes of the video because it's a really good explanation of what a half-life is and how you determine how old an organism by how much of that radioactive isotope is left. And again, just like in chapter 20, when I said we'd do a Hardy-Weinberg example in class, we will also do a half-life example in class as well, because you'll have to know how to do this on exam 4. So, but please watch just the first 2 minutes of this, and then we'll do one of those in class. So there's also, when we look for evidence of evolution, there's anatomical evidence by looking at certain features of organism, and we've talked about these before. These are called homologous structures, structures with different appearances and functions that all derive from the same body part in a common ancestor. So if you look at the bones in the forelimbs of mammals, you can see that now we are obviously have different functions, but the structures have all arisen from similar embryonic tissue. So here's a human, obviously our hand, the 2 bones in our lower arm, and then our upper arm, our humerus. Well, think about a cat or even a dog is the same way. They've got bones that we call phalanges, finger bones in their little paws, just like we have fingers. Now, their bones in their forearm are a little bit different, but their humerus is the same as well. So this shows a relative relatedness between the 2 organisms. How closely are we related? Well, how different are these bones? That will determine how closely we're related or not. Now look at a bat. Their phalanges, they've got them. They're way longer than ours are, but they have the same embryonic structures. A dolphin or a porpoise, same thing. And then a horse, now this is going to look a little bit different. Yes, they've got these same phalanges, but they have obviously developed into something different. But if you take a look evolutionarily at all of these bones, they all show some relatedness between all of these vertebrates. We also look at embryo evidence to see how closely related organisms are. The longer their embryos look alike, the more closely that they are related. So if you take a look at a human embryo, a chicken, a snake, salamander, fish, you can see some definite similarities. And of all of these, a human and a chicken probably look the most alike for the longer period of time. Right now in this picture, and it doesn't say, this is probably, I would say, maybe 8 to 10 weeks of fetal development. But look how similar chickens and snakes were. So reptiles and birds, we've always thought were pretty closely related. And look at these embryos, and that will show you. Salamanders, an amphibian, and a fish, we also know that amphibians and fish are closely related, and so their embryos look very similar as well. And then the last thing that we're going to talk about when it comes to looking at anatomical evidence for evolution is that even though we've got structures that are similar to other organisms, natural selection outcomes aren't always perfect. You know, we've talked about how natural selection makes one organism or one species more fit than maybe another species, but sometimes that's not always perfect. So if you take a look at vertebrate eyes and a mollusk's eyes, I mean, there are some similarities in eyes. Obviously, we each have 2 eyes, but here's something imperfect that natural selection did. Mollusks, their photoreceptors face forward, and it all has to do with how your brain perceives that. Well, that makes them not have a blind spot, which for them is really advantageous, especially in the depths of water when they can't, see around them too much. Well, unfortunately, our photoreceptors, for whatever reason, evolutionarily, this happened, they end up facing backward, and that's why we have a blind spot out to the outside of our peripheral vision, that blind spot where you can't see somebody come up behind you. So not all natural selective outcomes are perfect, and this is just one example. So that is the end of, that's just another picture of how they're all set up. That's the end of chapter 21, nice short one. And if you have questions, as always, let me know.